Hi, everybody. My name is Nina Bednarsek, and I'm a senior scientist at the Southern California Coastal Water Research Project. First of all, thank you so much for the invitation to present our work here today. This talk focuses on the integrated assessment of ocean acidification risks to sensitive pteropods in the northern high latitudes, ranging from the North Pacific Subpolar Gyre, Gulf of Alaska, Bering Sea, and the Amundsen Gulf in the Canadian Arctic. This study was supported by the North Pacific Research Board, with data also coming from the Ghost Ship P16 cruise and NOAA PMEL. This paper covers the work that has recently been accepted in Frontiers, special issue on OA and hypoxia in marginal seas. And today I'm presenting the results of our interdisciplinary work with all the co-authors here providing an important contribution to various aspects of this work. So for a short background, we know that the ocean acidification in the high latitudes occurs at the unprecedented rates and has already significantly changed the marine habitats. We also know, at least for a decade, that marine calcifiers are already negatively impacted in the polar regions. But since these marine calcifiers are economically and ecologically important, their negative impacts can have socioeconomic repercussions, either through food webs or ecosystem-related services. So as such, we need accurate, comprehensive, and interdisciplinary understanding and predictability of OA risks. To help delineate the risks, we're using the risk assessment framework from the IPCC. The risk assessment consists of the three components: the exposure, sensitivity, and the adaptive capacity. So under the exposure in here, we need to understand baseline natural variability, recognizing what is pertinent to a specific system. In this case, all the factors or stressors that impact subpolar and polar marine species. On top of that, climate change trends and projections, including extreme events that can non-linearly interact with OA, needs to be taken into account. And lastly, marine-based inputs like freshening and riverine inputs can play a substantial role in the polar regions. Defining the biological sensitivity, that's the second component, is complex, but we can try to strategically narrow it down to the most sensitive biological processes that have the capacity to induce bottlenecks and through this impact population level responses. In addition, life history matters because it helps determining when and where the exposure would be most severe, essentially providing an early warning predictor. And then the adaptive capacity, so that's the third component, can, if existent, leverage sensitivity and thus is that fundamental in studying along with the sensitivity. The process can happen across short or long-term scales, for example, adaptation. And in addition, some physical processes impacting spatial connectivity can define demographic processes and indirectly predetermine capacity of the responses. Okay, to the outline of today's talk, we'll go through three different risk assessment categories. So I will first present OA exposure across different regional scales that we investigated: North Pacific Subpolar Gyre with Gulf of Alaska, the Bering Sea, and Amundsen Gulf. Then we will focus on the biological responses in pteropods, including their shell dissolution, shell building processes, presence of different morphotypes. And lastly, we will discuss the elements of the adaptive capacity, including genetic structure, spatial connectivity, and try to bring it all together. Okay, so to characterize OA exposure in the North Pacific Subpolar Gyre and the Gulf of Alaska, we used observational P16N cruise data. This data shows that the corrosive waters are becoming shallower from 50° north onwards and reach biologically unfavorable conditions in the upper 50 to 100 meters. Now, looking at the GOA model, GOA cobalt model data from the Gulf of Alaska, from Hauri et al. 2020, there are several OA hotspots in the wider region. Omega saturation state is shoaling into the upper 50 to 100 meters and occurs during winter and early spring, partially also extending into the summertime. So this timing actually coincides with the spawning and the presence of the most vulnerable early life stages of pteropods. Now, to cover for the decadal changes, here showing a model output for the last 34-year period that suggests a significant decline of omega in the upper 100 meters, where the area of undersaturated water more than doubled since the 1980s. This means a significant reduction of suitable habitats for marine calcifiers. Moving to the Bering Sea, this is the Bering Sea 10K model output that suggests unfavorable conditions for pteropods in the subsurface water between 50 and 100 meters across nearly all of the Bering Sea shelf. There are even more severe conditions in the northwestern shelf for both winter and summer, which means that early life stages as well as the adult pteropods are exposed to less favorable conditions. Now, for the Amundsen Gulf, the undersaturated conditions occur in the Pacific halocline layer between 50 and 200 meters, depending on the location. Carbonate chemistry for this region was recently described in Niemi et al. If you want to check for more details. And pteropods' main habitat in here is largely corrosive in summertime, where so far northern pteropods are still in the early life stages. All right, now going to the biological sensitivity, and here introducing pteropods as OA indicators. So pteropods are shelled pelagic snails and belong to zooplankton group. They're essential food source for salmon, Arctic cod, char, especially in the fall period where the rest of the food is already absent. Their shell is made of aragonite, more soluble form of calcium carbonate, and it's exactly because of this rapid shell dissolution that they are an ideal sentinel species for oceanification monitoring. However, we currently have limited understanding of OA impacts on pteropods in the polar regions, especially in the north latitudinal regions, and related to their sensitivity and adaptive capacity. So the first response we investigated is shell dissolution. Pteropod shell dissolution was observed in all ocean basins across the investigated high latitudinal habitats. We found the most dissolution in the Amundsen Gulf, followed by the Western Gulf of Alaska and the list over the Bering midshelf. And the dissolution was closely correlated with the carbonate chemistry. So for example, here on the left in the Subpolar Gyre, the greatest dissolution co-occurred with the shallowest aragonite saturation state. For the Amundsen Sea, Niemi described various processes contributing to the dissolution. And interestingly, we also found shell repair in pteropods. However, this was only the case for a very limited number of organisms and always the adults, so not early life stages. In this case, we strongly think that this cannot contribute to the pteropod resilience on the population level. The second response we investigated were shell building processes. So another process that can tell us something about the biological sensitivity or their adaptation scope. So in this region, there are several morphotypes present, so different shapes of pteropods. We observed that at high omega, pteropods build the shells with inflated spire, while at lower omega, the shells has depressed spire. This could be a potential adaptation as flatter shells has less surface exposed to corrosive conditions. However, we also conducted X-ray micro-CT measurements to investigate their shell building capacity. So we essentially measured shell thickness as a metric of calcification. There we found that their thickness declined with lower omega state with a threshold of omega at around 1. And such lower calcification has all sorts of repercussions, including increased predation, energetic constraints, also biological pump. So we estimate that the sensitivity prevails here over the adaptation. And the last component here is the adaptive capacity. First, let's take a look at the genetic structure. We investigated genetic structure based on the cytochrome oxidase 1 mitochondrial DNA locus, and we found low haplotype diversity with no genetic differentiation between different morphotypes. The minimum spanning network described several short branches connected by a common haplotype, and this can be often interpreted to represent an evolutionary recent founder event followed by the population expansion. So even on a larger regional scales, from the west to the east-north Pacific to the Svalbard and the Kara Sea, most haplotypes differ from each other by only a few mutations. In general, this lack of genetic structure across large scales indicates reduced potential for the adaptive capacity. And one more thing that we can say with greater certainty is that the morphotypes present are the results of the phenotypic plasticity and not genetic differentiation. And the last component includes spatial connectivity. To determine connectivity, we conducted particle tracking in the Subpolar Gyre in the Gulf of Alaska and the Bering Sea. Blue colors here indicate shorter, and the red colors indicated longer time intervals since their release, which is indicated by the black squares on the figures. And so what we observed is that in the Gulf of Alaska, pteropods are supplied via this northwards advection in here. There is a high level of spatial connectivity throughout their life history, and this indicates some mitigation against OA. However, the supply of early life stage pteropods originate in the OA hotspots, and that to a certain extent counteracts the mitigation capacity. And on the other hand, the spatial connectivity in the Bering Sea is much lower, so there is much less potential to avert negative OA impacts. So in conclusion, in this work, we have identified regionally specific risks related to OA. There are unfavorable conditions found within the vertical habitats across all regions. There's also pronounced seasonal patterns with low OA coinciding with spawning events. And based on the all integrated results from our study, pteropods are at high risk despite some mitigation parameters found. The application of OA risk methodology we used in here can be used for various pelagic species with similar sensitivities in their respective habitats. Such comprehensive risk integration as conducted in here permits improved prediction of ecosystem change relevant to effective fisheries resource management. And we found that pteropods are an excellent indicator paving the foundation for future OA monitoring for the ecosystem health. And that information can be used for a variety of different stakeholders in the polar regions. We also demonstrated how the various field programs can be integrated, like ghost ships and regional multi-year cruises in the Bering and Amundsen Sea. All right, we're at the end. I wanted to thank you for listening. Here I also provided the link to our accepted papers on this topic. I also wanted to thank to all the people who helped in this work to be completed, and see you at the live session.